So our next speaker is Manu Leonetti from CZI Biohub, and I think going to talk about intracellular map of the human proteome.
Yeah, thanks, Meg. It's really a pleasure to talk right after Jonathan because some of the work that he's done years ago in East has really been a huge driver and inspiration for some of the work that I'm going to present. So my lab is really interested in developing tools and data sets to understand how proteins are wired in our cells and how this wiring drives cellular functions. But at a high level, what we're trying to achieve is what I'm calling cell biology at the scale of the whole cell. In other words, how could we study any cellular response, for example, the activity of one of our favorite signaling pathway, but really in the context of the cell as an entire integrated system? Can we study all the different correlation of that response with different parts of the cell, so on and so forth? And of course, this is a very complex problem. And as Aviv alluded to yesterday, the cell is so complex that we cannot achieve measuring every single thing that we would love to do, right? Then to me, to achieve cell biology at the scale of the whole cell, this idea of being able to capture global cellular profiles, right, measuring organization and activity across the entire system, becomes an exercise of dimensionality reduction. In other words, how could we reduce the complexity of the cell, and Jonathan was just speaking to this just before, down to a manageable number of measurables, right? And then the question becomes, what should these measurables be? And so what is the kind of compressed panel of measurements that we can do that give us a critical coverage about cellular functions? I think that this question is central in cell biology today, and especially central in the age of AI, because we're going to need ways to prioritize the kind of data that we should produce to get the very high amount of training for all the models that we're dreaming of developing. So I want to discuss this problem in two aspects. I want to first give you an example of how we solve this problem in a very specific context. And then towards the end of my talk, I'll go back and have a more conceptual discussion about how to generalizing thinking about this. So one way that we approached this problem was to deconstruct the cell at the level of individual organelles, and these organelles become the compressed panel that I was talking about. What these experiments look like are that we're using CRISPR to tag organelle markers in an arrayed format. We're using tags that have both a fluorescence marker and an epitope tag. So we're going to tag the mitochondria in one batch, the Golgi in another, so on and so forth. And we can use these cell lines to study how organelle morphology changes as cells undergo perturbations. And I'm not going to be talking about this too much today. But we can also use these cells and study how the protein composition of these organelles is changing using mass spec. And specifically here, what we're doing is we're breaking the cells open by very gentle mechanical lysis. The cell is exposing its guts, but all the organelles are still intact. And we can come with antibodies against the tag, immunoprecipitate these entire organelles, and send them to mass spec. So this was really a fantastic collaboration between many different people in the lab, driven in particular by Mark O'Hine and Duo Pong. And all the mass spec has been done in very close partnership with Josh Elias at the Biohub. And all of this has been published. The paper just came out literally 3 weeks ago. So we did not invent this idea of doing a native organelle immunoprecipitation, but classically, people have been looking at one organelle at a time, mostly mitochondria and lysosome. And the idea that Marco, Verena, and Frank had, which they were really driving the experimental part of this work, was how about we apply this method to all the different organelles that we can think about? And so what they ended up doing is coming down with a list of 37 protein markers that cover all the different compartments, both membrane-bound and membrane-less, that we could think about, including a few big assemblies in the cytoplasm, for example, the translation machinery, and really applied this native IP workflow to all of these things at the same time to, again, get a complete cellular profile. It turned out that this worked really well. Surprisingly, this also worked really well for membrane-less organelles, and I'd be more than happy to tell you more about that. All of this is in the paper. Also, something that was really interesting is that a lot of the contacts that we know exist between these organelles inside the cells were still preserved to some extent in the pull-downs. And we know, for example, from the work of Jennifer in particular, that organelles make extensive contacts with each other. What I'm showing you here in this heat map is an enrichment plot where all the different organelles that we pull down on are here on the y-axis, and on the x-axis are the proteins for different compartments that we see enriched in all of these pull-downs. You can see, for example, that peroxisome and mitochondria are really co-enriched for each other, and that makes complete biological sense because we know that these two organelles really have a lot of biological feedback with each other. And what's really cool is when we pull down on the ER, for example, which is really the master organizer of membrane organelles in the cell, we know that the ER touches pretty much everything. Well, when we pull down on the ER, we actually get enrichment for all the different membrane-bound organelles from lysosomes all the way to mitochondria. So what we're left with at the end is a very rich data set of protein enrichment. We have coverage for about 8,000 to 100 proteins that, we think that there's at most 10,000 proteins expressed in the cells that we're looking at here. So we have coverage for almost the entire proteome. And each protein is going to be defined by its enrichment in these different pull-downs, which you can think of as essentially enrichment is in targeted fractions of the cell. And Duo and Kibom were thinking a lot about how to approach the analysis of this data set. And what they decided to do at the end was to borrow a playbook from single-cell RNA-seq analysis, where we transform this very large matrix into a graph. So this is one of these nearest neighbor graph. Specifically, what we're doing here is for each protein, we're identifying the 20 other proteins that share the most similar enrichment profile. And we're going to connect all these different proteins at the level of a graph, and we iterate that across the entire data set. And it turns out that these KNN graphs, especially in the context of this kind of data, we think has a huge advantage of denoising all of these signatures. And on top of that, of course, graphs are very amenable for clustering-based analysis, which allowed us at the end to identify territories in the graph and identify organelles. So the final product of this analysis is a spatial map of the entire cell, which I'm showing you here in a UMAP format. Each dot here is one protein in the cell, and we color-coded them for different organelles that they belong to. What's really satisfying is that the structure of this map makes complete biological sense, especially the entire secretory pathway is organized here from ER to Golgi to endosome to plasma membrane. What I haven't shown you is that the definition of these labels, the colors here on this graph, was done in a completely unsupervised way. So what we did is we did pure clustering. We identified the clusters, and you're going to have to take my word for it. It turns out that the boundaries between different clusters defined in a completely data-driven way correspond beautifully to the exact boundaries between different organelles. And if you want to know more, you can check the paper. All of this was done in a transformed cell line, HEC293 T cells, because we needed to start somewhere that was easy to do, but I fully share Jonathan's sentiment. We want to move away from these transformed cell lines, and we're really pushing all of this workflow into iPSC models and especially differentiated cells. We also have a website at organelles.czbiohub.org where you can look at all of our data. There are really two things that I love about this approach. The first one is that this map is inherently interpretable and actually contains resolution that extends far beyond just organelle membership, but all the way to protein complexes. So remember, underlying this map is a graph. So I'm showing you here a little piece of this graph at the interface between endosome and Golgi. And I color-coded a few different protein complexes that we know work together to move cargo between these different places. Things like the WASH complex, retromer, and commander. If you're not a cell biologist, it doesn't really matter. But what's really interesting here is that even in this coarse-grained description of the cell, a small number of measurements, we still have enough resolution to identify and predict to some extent protein complexes and how different complexes work together to perform separate functions. The second thing that I really love about this approach is that by design, it's pretty simple to do. In the original map, I was telling you that we tagged 37 markers. Well, in subsequent analysis, we show that we get most of the information with 15 markers, and so that boils down to a set of experiments that's really easy to scale and to deploy across multiple conditions. And of course, what we really wanted to be able to measure is how the entire cell gets rewired as cells undergo a perturbation. We decided to start with infection with a virus. OC43 is a coronavirus in the exact same family as SARS and SARS-CoV-2, but we're all immune to it, and so it's much safer to use in the BSL-2 lab. And so we infected the cells, and what Duo did is he got two graphs, one in uninfected conditions, one in infected conditions. He used data analysis tools to align these graphs in graph space and essentially identify proteins that relocalize between the two conditions. I'm showing you here in a UMAP format. Again, here, each dot is one protein in the cell. This is color-coded for different compartments in an uninfected map. You can see that for the first time, we can do the experiment that we really wanted to do at the onset, which is to capture how the entire architecture of the cell gets remodeled during perturbation. This is viral infection. We're really throwing a bomb in the cell here. So about 8% of the entire proteome gets relocalized, so to speak. It turns out by being able to identify proteins that relocalize, we really got a completely new window into the cell biology of this virus. What was really striking to us is that there was very little overlap between the list of proteins that seem to relocalize during the process and proteins that change in abundance, as if cells are using these two modes of regulation in a completely orthogonal way. And there are other examples in the literature using completely different perturbations that suggest that this might be actually a fairly universal theme. Relocalization and abundance regulation being used in an orthogonal manner. Of course, we can do gene enrichment analysis on these different groups of proteins, and they tell us completely different pictures of what's going on and complementary pictures of what's going on in these cells, where abundance changes being mostly related to innate immune responses, but relocalization really being heavily enriched for things related to homeostasis and stress response. Something that did catch our eye was pheroptosis, which is a form of iron-dependent programmed cell death. We followed up on this a little bit, trying to understand its relationship with infection. Chad and Maduria really wanted to dissect this a little bit more. They could show that indeed, using a dye, pheroptosis is activated during infection, and we can suppress that by chelating iron. But even more interestingly, they showed that pheroptosis is both necessary, but also a driver of infection itself using pharmacological experiments. When they inhibit pheroptosis, they see a decrease in infection. You can tell me like, well, okay, fine, But what's really cool, I think, is that we can activate pheroptosis using the drug, and we see an activation of infection. And it's actually very hard to make a virus work better than it actually already is. So I think that's really telling us something fundamental about a specific role of pheroptosis somewhere in the life cycle of this virus. Our favorite hypothesis, there's a metabolite, probably a lipid downstream of the stress pathway that the virus needs either to make its replication machinery or to make its capsid itself. And we're trying to follow up on it. Okay. So I hope I convinced you that this one solution of reducing the dimensionality of the cell, and here looking at the composition of different organelles, is an interesting way to get global cellular profiles, and more importantly, really illuminated for us some really fundamental cell biological mechanisms. And there are many advantages to this method, including this molecular level resolution that I just mentioned to you. But of course, there are some really, really serious limitations to this, right? And in particular, there's no way we'll ever be able to do this at single cell resolution. So everything that I've shown you was population average. And of course, this is absolutely not compatible to looking at cells live, in situ, and in vivo, which is definitely, especially in the context of 4D cellular physiology, where we all want the field to be able to go. So then, for the last 6 minutes here, I'd like to discuss just conceptually how we could achieve checking all of these boxes. And of course, the one modality that potentially has all the answers is imaging and life cell imaging in particular, right? We can label things in cells. We can use different kinds of label-free method. This is a quantitative phase, for example, to get very, very deep profiles of cellular architecture and cellular function. I think we all agree that, cell biology is re-entering an age of imaging in terms of phenotypic power. But I think there are then, in this context, two key questions that we should be asking. The first one is, well, how are we going to choose what to image to maximize the encoding of information in these image assays? Right? What is the solution, especially if we want to do these experiments in live cells, where the number of channels is going to be very, very limited? What are we going to choose to image? We heard a few possible solutions this morning, right? Caroline could tell us, like, well, let's image chromatin, right? And GN could tell us, like, well, let's do Raman, for example. I think these are both fantastic possibilities. I'm advocating for a very systematic and quantitative evaluation of the information power of these different modalities side by side, so we know how to make the best choice. And the second big question is, well, it's great to be able to recognize that something changes, but of course, as cell biologists, what we really want to be able to do is interpret function. And so my dream is that we would be able to find a compressed panel of imaging features that could include label-free morphology features or reporters, but a small enough panel that we could really deploy that, especially in live cells, or maybe in the future in a live animal, and build an AI interpreter that can translate this information into a full functional signature of cells. What I would like to know is I look at this cell, and I can tell that it's in this phase of the cell cycle, it's glycolytic, and it's doing mTOR signaling. Right? I think this is the level of interpretability that, as a cell biologist, I really would like us to aspire. I don't have a solution for this, but obviously we are working on this, and I really wanted to start a conversation with all of you. But I'm really hopeful that this problem is solvable. And let me give you just a few snippets of why I think that's the case. Well, first, I think we have all the technologies to get the training data to solve that problem, mostly because we can image cells, see how they look, and at the same time measure very deep profiles about their molecular states, doing, for example, spatial transcriptomics, measuring their metabolism using imaging mass spec, or their activities using biosensors, right? So I think from a technical perspective, we should be able to have access to the right tools. The second thing that makes me hopeful is that we know that biological images have this uncanny superpower to really encode a ton of very specific functional information about cells. One example of that from the literature that was already discussed previously are these OPS screens. There's one paper in particular that really inspired me, this paper by Paul Blaney and Ian Cheeseman a couple years back, where they were just imaging 4 generic markers in cells, DAPI and cytoskeletal components, essentially, right? So a very small version of cell painting. They were doing CRISPR knockout screens on all of this, and then there were, in the classical analysis, clustering genes, gene knockouts that shared the same phenotype, and essentially they were identifying more or less the exact same clusters that Jonathan was identifying in his lab during genome-wide perturb-seq, right? And so I'm being provocative here, but it looked like there was almost as much phenotypic power in these 4 markers image with fairly classical microscopy as the kind of signatures you can get from entire transcriptomes. Another example driven from our work was analysis that we've done from a data set that we produced a couple of years back called OpenCell that you might be familiar with, where essentially we were using CRISPR to build a GFP tag library in human cells. And you can check our data set online. We have data for over 1,300 proteins in human cells, and this was all live cell confocal microscopy imaging and mass spec protein-protein interaction data. But using all of this image data, one of the questions that we were really excited to address was, what kind of functional relationships can we infer between 2 proteins just by comparing their subcellular localization? To do that, we were super lucky to collaborate with my colleague Loïc Royer at the Biohub and his postdoc Hero. And together we built a self-supervised deep learning model to transform the information in these images into latent space, and a vectorial space in which we could do math and clustering, so on and so forth. All of this is published, so I'm not going to go into the detail, but let me show you some of the results that we could achieve with this. So I'm showing you here the result of a clustering that we did in latent space. Here on the x-axis, we're increasing the resolution of clustering, so we're breaking down the data set into smaller and smaller clusters that group proteins that share together finer and finer similarities. And on the y-axis, we're comparing the definition of the clusters with essentially ground truth lists. And I'm just going to say that all the values here are really pretty high. So what the model is capable to do in a completely data-driven way is recognize, learn organelles at low resolution. Okay, that makes complete sense. I just showed you the images. What was really surprising to us, though, was that as we were increasing the resolution, we could identify with almost the same precision functional pathways all the way to protein complexes. That gave us the idea that we could just compare these subcellular localization images and predict new protein-protein interactions. And we did that. We took a protein for which nothing was known. We imaged it. As you can see, this is an ER protein. We have a lot of ER proteins in our data set. Yet we fed this image to our machine learning algorithm, and it came up with one very specific hypothesis, which was that this protein was likely to be a new subunit of OST, which is a glycosylase at the ER membrane. And lo and behold, we did the mass spec and we verified this. Right? And I just want to point out that this is not an intuitive result to me, right? These are diffraction-limited images that essentially contain information at the micron scale, and protein interactions are nanometer scale relationships, right? So here we have 3 orders of magnitude conceptually of difference in biological resolution that yet image analysis can bridge. So I hope I convinced you through all these examples that I believe it is possible to build an image to AI interpreter, and that then the questions that we need to ask ourselves are, what exactly in cells are we going to choose to image? And I think it's likely to be a collection of morphology features and biosensors. And how can we very quickly diversify cell states at the level of training to push cells in different corners of their phenotypic landscape and really start training models that learn generalizable rules. And I really think that these OPS screens offer really, and the kind of screens that also Jonathan was talking about just earlier, really offer a really cool possibility. So at the end, and especially if we can learn to do all of this with live cell image features, we could look at cells over time, really understand the succession of activities going on in these cells, understand how they process information, how they respond to input, and how they make decisions about their future in the context of cell in a dish, an organoid, and I hope tomorrow a whole living organism. I'm going to leave you with all these ideas. Let me give a giant shout out to my team and my collaborators, and I'll be happy to take some questions.
Yeah, hello. Yeah, so great talk. So I have one question related with your multi-resolution clustering. So I think even with the best results, so the random adjusted index is around 0.5. So I think that probably there's still lots of the room to just improve. So do you have maybe
For
kind
sure.
of
that's a great question, and that's also related to the metric that we used here to measure the quality or the precision of the clustering, ARI, is great, but it has a lot of limitations. And especially for ARI to be really high, you need the boundaries. ARI is very sensitive to even a very small number of like miscalls in your classification, which I think is not a fair assessment of the complexity of the data here. We're evaluating now. So we think that ARI, it's a great kind of like first pass metric to assess clustering performance, but we actually have moved to like a little bit more precise metrics from now. Yeah.
Hi, great talk. So just riffing off what you were just saying and the classifiers by AI, have you tried just looking at the markers for the organelles and comparing relative distribution for different cellular states to see if like a trained program, for example, will be able to identify like a fingerprint, but based 100%. On the organelles.
And I think we're going to hear from Daniel about something very similar right after. And I think organelles are a natural first candidate set, right? The question is, and frankly, and I was discussing that with Jennifer over a coffee break, it's really likely not to be not to be enough, right? So how do we move beyond that? And I think morphology is going to tell us, a lot. But at some point, if we really care about measuring protein activity, we're going to need to put biosensors. And I think some of the sensors that your lab has been developing are actually amazing candidates, right? Then the question could become, well, how can we pack, multiplex biosensors in the same cell to be able to do all of these, high-dimensional phenotyping in a live cell setting, for example?
I was wondering, does one need to consider several time points when they want to look at the proteomics after a pulled knockout? Because there will be a time for turnover. You might not catch the effect if you don't hit the right time.
You're absolutely right. And actually, new experiments that we're doing in viral infection, we're actually taking multiple time points during the infection cycle. To start addressing some of the questions that you're thinking about. I absolutely agree with you. Then, that the power here of these approaches is that, again, they're not super complicated to do experimentally. And so we can start multiplying, the number of samples that it would take to start having more temporal resolution.
And then do you prefer the one that you start seeing changing when you feed your model? So which time point you're going to pick? Yes,
well, if you want to feed things to your model, you don't have to choose, actually, right? You can put everything and the model can sort things out. Yeah. Okay. Thanks for the question.
Thank you, Manu. That was a really visionary talk. And I want to add fuel to your fire by asserting that basically I think that future is not only possible but inevitable. Mm-hmm. And my perspective for that comes from the machine learning field, where there's a concept called the bitter lesson, which is learned often in different fields, where expert systems and expert intuitions often get replaced at an inflection point where data and computation can take over. The key thing I think is missing that the field needs to define, and maybe you and others can weigh in on this, but we have lots of time to talk about it today, is like, what is our metric for progress? Like, when do we know that we've arrived? I think once that is very clear and metrified and quantified, I think it could spur progress. But yeah, thank you.
I completely agree with you in defining clear goals for these models in particular. And I imagine this is something we're going to be discussing at the panel later today, right? The way I think about it, to me, if a model really understood what a cell is and how a cell processes information, the key prediction should be to start with a movie and predict where that cell is going to be at some point in the future. That would be the first order, test that I think would really really challenge the models to have understood something profound. So,
let's take a Zoom question then. Yeah.
Kuntin Mielke made a comment that Mito are definitely doing their own thing in the UMAP in your organelles section. I think that highlights maybe a question of connections that were absent. Mm-hmm. In this, like you showed where things made sense, but yes. I think there may be some gaps in knowledge as well, or gaps in connections in that.
For sure, there are some gaps in connection. Well, first of all, you have to take the UMAP with a big grain of salt because it's a very imperfect representation of the whole data set, right? You can't capture everything just in 2 dimensions. The best way to really look at the connections is to look at the graph itself. And in that website, organelles.czbio.org, we let the users look at the graph. So there might be some information that's already in our data that can be approached there. But of course, there are still a lot of things that we're missing from this analysis. I'll just mention one. as Emma was talking about this morning, a lot of proteins might localize to a lot of different compartments and things like this. Right now, especially in the way we're labeling the localization of these proteins, we're only labeling one compartment for each protein, and that's really imperfect. And we're really trying to, it turns out like our assays make it a little bit harder to evaluate and quantify multi-localization, but we're developing new models to start to be able to annotate these cases. Yeah.
I'm curious about imaging with biosensors. Are you imagining targeting different biosensors to different organelles? And with that, what types of challenges are there? Because different organelles have different microenvironments, etc. So I'm just wondering whether you have thought about that. And then kind of a follow-up to that question is, most people look at sensor response as kind of on and off, but I think there can be rich data in the dynamics or the pattern of the response. And if you have thought about that, I would be curious to hear what your thoughts are.
These are two great questions. In terms of multiplexing, or in terms of imaging sensors, what I'm thinking more about is multiplexing a lot of sensors in the same cell. What you're talking about, I think, from a cell biology perspective, is super interesting, right? And there's beautiful work that's being done in neuroscience, for example, looking at a localized metabolic activity in different parts of the neuron. And actually, Daniel was asking a question earlier, doing some cool work on this. And so 100% yes to the power of localizing sensors to different parts of the cell to understand subcellular metabolism. I think that might be a little bit too zoomed in for some of the high throughput phenotyping that I'm calling for. But yeah, I hope that answers your question.
Oh, sure.
And sorry, I did not answer the second part of your question because I already forgot it, but maybe we can discuss that over coffee break.
Sounds good. Yeah.
Thank you, Manu. I think this is an interesting time. We can continue discussing the panel session. Yeah.